Welcome to today's USP PharmaTech webinar, Applying Process Analytical Technologies to Support Real-Time Release. Thank you for taking the time out of your busy schedule to join us. Each registration is unique and grants the right for the individual registrants only to view this webinar. Sharing this webinar with unregistered viewers either during or after its performance violates USP copyright and is prohibited. Next. If you have not done so already, we ask that you take some time to read the USP Education Course Disclaimer. You can download this presentation, including the disclaimer, from your USP Education account. Please follow the instructions in your confirmation email to download the presentation. Next. Presenting today is Brian Glass, Senior Analytical Consultant at PharmaTech Associates. I will now hand the presentation over to Mr. Glass.
Hello, everyone, and welcome. Today, I would like to take a minute and discuss some of the benefits of adopting a quality by design approach and one significant benefit of this approach, which is real-time release testing. Of course, this has the obvious benefit of reducing testing through your in-house analytical laboratory, which is going to save time and increases the speed that you can get to market for your new drug products. In this webinar, I really want to focus on oral solid dosage manufacturing, primarily because this is still the predominant dosage form that we find today. And from a continuous manufacturing perspective, oral solid dosage forms are well suited for this type of analysis. Recently, there have been many advances in continuous manufacturing of other dosage forms, as well as new product analytical technologies that are currently being developed for API manufacturing, oral solutions, and biologics. But for today, I want to focus on oral solid dosage forms. A thorough understanding of the manufacturing process allows for the identification of critical material attributes and critical control parameters to ensure that critical quality attributes of the finished product are achievable. To ensure this, process analytical technology, coupled with chemometrics and multivariate analysis techniques, can be used as tools to generate data demonstrating control over the process. One of the drawbacks of the batch manufacturing process, and a batch manufacturing process can still utilize process analytical technology, but if you've got a basic batch manufacturing process and no PAT in place, a big drawback is the length of time it takes you to produce a batch. Often this involves multiple steps and holding times. You might start with blending, and then the blend is then segregated into containers which have to be sampled, stored, and then the samples sent to the analytical lab for testing. And reporting of the data concerning the quality of the intermediate often has to be held, you have to wait until that testing is available before you can continue to the next step. Most of the data available that I'm going to talk about today describes real-time release testing, more or less in the context of a continuing manufacturing process. I do want to stress that a batch manufacturing process can also incorporate process analytical technology and therefore real-time release testing. So this slide really shows you where process analytical technology comes into play. During the actual processing, you can collect data, and that's properly coupled with the correct statistical analysis and the delineation of a design space can allow you to proceed quickly from lab scale through tech transfer, and you have continuous information that is constantly being captured regarding the quality of the batch. So this quality by design model is differentiated from the classic batch manufacturing process by the determination of a design space. And this is a space which delineates or maps, if you will, the adjustments in the process that can be performed to ensure quality in the finished product. Your standard batch manufacturing model uses a fixed manufacturing process, but raw material inputs may be variable. They may be from different manufacturers, different lots, etc. And this is, of course, going to lead in variability in the finished product. But in a quality by design approach and utilizing this approach, you can still have variability in your starting material. But if your manufacturing process is adequately controlled, and we can do this with our process analytical technology tools, we can ensure a consistent finished product. And this is the big goal for regulatory authorities. They really want to make sure that the finished product is consistent in its quality. That comes up time and time again. Now, this slide is showing the predominant routes used to produce oral solid dosage forms. And usually the three big ways that these are manufactured are through either direct compression, where you take a blend and it goes directly into a tablet press where the tablets are formed. You can also use a dry granulation or a wet granulation process. The overarching goal here is really to utilize PAT tools in order to monitor the process in real time. And while you're doing that, you are collecting data to demonstrate that the process is under control. And the data generated during this processing can be used to release the final product without the need for any additional laboratory testing. So I'm showing here some of the most common process analytical technology tools that are currently used. And these are very common in a continuous manufacturing process, but you don't have to adopt a continuous manufacturing process in order to utilize these tools. In the case of a direct compression, you would have raw materials going into a blender, and then those go directly to a tablet press where tablets are formed. Probably one of the most common PAT tools you're going to find are Raman or FTIR, which is used initially to confirm the identity of the raw materials. Near infrared spectroscopy, very useful for determining blend uniformity. And content uniformity as well of the finished product, in some cases, can be determined using near IR spectroscopic techniques. In a dry granulation process, you're typically going to go, and I picked roller compaction. This is just probably the most common, but there are certainly others. This sort of a dry granulation produces ribbons, which can then be milled. And depending on the milling, you could use near IR again for not only blend uniformity, but for your moisture content. You can also use particle size distribution. We do have these tools available that can determine particle size distribution. And then content uniformity again by near IR. And then finally in the wet granulation, where you're going to take these blends into a granulator with a liquid, drying is going to be important. So near IR is commonly used here. Again, particle size distribution is important, and you can determine content uniformity of each tablet as it's coming out of the tablet press. So the pad tools that I'm describing here can be inline tools, which is a probe that is placed in a specific point in the manufacturing equipment that continually monitors and giving you process results in real time. You can have them online where a portion of the batch is segregated and then tested very near to where the manufacturing is occurring. Or it could be at line or offline where samples are taken and sent to the lab. The initial step in a tabling formulation is always going to be blending of the API and excipients until a reasonable homogeneity has been obtained. Often, in just the batch manufacturing process, determining blend uniformity, which is a critical component for your process validation, often this can be problematic to do manually because the physical act of collecting samples for analysis can result in either segregation or desegregation of the blend at the point that you're actually doing the sampling. Usually these local effects can be produced by the introduction of a sampling thief. And you can eliminate this sort of issue if you use process analytical technology during the blending process. Likewise, for wet granulation processes, moisture content is always going to be a critical control parameter that affects the quality of the finished product. It's important to know exactly how much water content or moisture content you have. This can be captured on the fly and makes it a lot easier rather than having to sample and run the risk of this sort of a bias in your sampling, and that then has to be sent to the lab for maybe Carl Fisher analysis. And then, of course, likewise for roller compaction and other dry granulation processes, ribbon thickness is often critical. And pharmaceutical scientists have been making really good progress in developing non-destructive technologies to capture this data during the processing, that is on the fly, if you will. So PAT sensors can broadly be categorized into two big areas. There are hard sensors and there are soft sensors. A good example of a hard sensor could be an FTIR or Raman probe for raw material identification. A lot of times the hard sensors that you're going to see use spectral signatures or spectral attributes that are unique to a particular compound that when properly calibrated can provide both qualitative and quantitative data regarding the compounds in question. A soft sensor requires measurements from several hard sensors that, when coupled with an appropriate mathematical model, can predict a critical quality attribute. And of course, these are very important because you want, in a quality by design approach, you want to make sure that your critical quality attributes are being met to ensure the quality of the finished product. So probably by far the workhorse of PAT probes that you're going to see these days are going to be either FTIR, Fourier transform infrared spectroscopy, near infrared spectroscopy, and or Raman spectroscopy probes. These are usually hard sensors that, as I mentioned previously, are used to examine spectral signatures of the different compounds to kind of positively confirm their identity and amount. Also, water has a fairly unique spectral signature, and it's commonly used to determine the water content, as we discussed how important that was for the wet granulation process. All of these techniques are kind of lumped into what chemists call vibrational spectroscopic techniques. And the focus really is on evaluating the absorption or transmission of specific wavelengths of electromagnetic radiation to provide a unique fingerprint for identification. So in this case, we can see here that, for example, any of these techniques could be used to determine identity, assay, blend uniformity, content uniformity. And all of this data can be used to substitute, if you will, for finished product release testing in the either quality control or analytical laboratory. Well, these are the workhorses. There are several other probes and techniques that have recently been described in the literature that offer a little bit more detail. And I find them a bit more interesting. So let's take a closer look at some of these technologies that have been out there, that are out here. So in addition to near infrared spectroscopy for water determination, MRS or microwave resonance technology has also been successfully used to monitor water content during a fluid bed drying and or a wet granulation process. So basically, these sensors can detect water during the actual formation or coating of particles that are produced through a wet granulation process. As a soft sensor, this probe's also been shown that you can take this data and correlate it with granule size. And that could be very important because a lot of times granule size directly determines your actual dissolution characteristics. And dissolution, of course, is a very critical component of oral solid dosage forms. And so it's nice to be able to have this data to support an application that suggests that we don't necessarily need to do dissolution testing because we already have the data that says, and the model that this will be fine. Drag force flow sensors. These are really cool devices that measure the flow characteristics of a wet mass consistency that you're going to find during, for instance, like a high shear wet granulation process. So basically, this is like a little finger that as material flows against it, the sensor is deflected. And that difference or that angle can be detected with optical strain gauges, and that can be correlated directly to density changes in a wet granulation. And of course, the density of a wet granulation is going to affect particle size, porosity, etc., which are going to directly impact your finished product properties. Again, primarily, especially dissolution. That's the first thing that comes to mind. Acoustic emission spectroscopy has been used in the past to quantify properties such as tablet thickness, mass, and other physical properties of the finished solid oral dosage form. Terahertz pulsed imaging and terahertz time domain spectroscopy have been shown to be very useful for monitoring the thickness of a film coating, as well as pore size in coated tablets, as well as granules. So these techniques use terahertz energy pulses, which I like to think of as far infrared. I mean, we've talked about the near infrared and the infrared, but there is a terahertz that's kind of the far infrared that can determine the essentially the effective refractive index of a tablet. And that can be deconvoluted to determine porosity, pore shape within the tablet. The only problem with this technique is that you usually have pretty low resolution and some long measurement times. And we'll talk more about measurement times, well, as it concerns or relates to process analytical technology a little bit later, but that is a very important aspect that you need to consider when selecting process analytical technology probes. We've talked a lot about particle size, and two of the biggest, probably the most important technologies that can be used to determine particle size during the physical processing are the focus beam reflectance and spatial filter velocimetry probes, where essentially particle size and shape can be determined using these sorts of measurements. And a laser in, for instance, a focus beam reflectance probe scans back and forth across the sensor and backscatters some of the light. And then that light, that backscatter, as it passes through, can be used to determine the chord length of particles, which is really a fundamental measurement of particle size. And in a very similar manner, they're almost inversely related. Spatial filter velocimetry also provides particle size information, but in this case, it's the shadow of the particle that passes through the laser that determines the chord length. So that's more of an indirect, but still valuable and technology. Optical coherence tomography. This is like an ultrasound, except instead of sound waves, you're going to use light. And using that, you can image internal structures that can provide you a lot of data on what the internal structure of your tablet looks like, and thus what you can expect the critical quality attributes of that finished product to achieve. So I think the big point here to understand is that process analytical technology is really a moving target. And both industrial and academic pharmaceutical scientists continually publish research in this area all the time. And just to go a little bit more deeper into particle size, because it can be such a critical part of your finished product, and it can affect so many critical quality attributes. Particle size and porosity are of a particular concern if you want to use your process analytical technology data for surrogate dissolution modeling. And by that, I mean, it is possible to use the characterization of your finished dosage form in order to convince regulatory authorities that you don't have to do dissolution every time. It is a lot of work. It won't work for every product, but the chord length distribution is probably, in my opinion, the most common technique that's used in order to determine a particle size distribution. Also, porosity of the tablet. Porosity measurements have been shown to be very helpful for developing a model for dissolution real-time release testing. Several products have been approved that use this modeling instead of relying on manual dissolution testing. I'm pretty sure, I believe all of these products have been immediate release, but research is continually progressing in this area. You can expect to see more and more interesting data, and but just to note that surrogate dissolution testing is not going to work for all of your products. But if you can utilize this technology, it certainly would save a lot of time if you didn't have to perform dissolution testing on every single batch. So this is a kind of a summary of some critical quality attributes and the process analytical technology that we've discussed and how they can be used. For blend uniformity, near IR, Raman spectroscopy, moisture content, magnetic resonance spectroscopy. If you're doing a wet granulation / fluid bed drying process, this can be done very, very simply. Granule size, of course, ribbon density for a dry granulation process like roller compaction, the strength or friability, if you will, of the tablets, as well as their porosity and coating thickness. All of these things we can nail down with process analytical technology. And based on that, we can reduce the amount of time that it takes to test these in the lab. We can release them using our process analytical technology data. We do not have to take another sample and then send to the lab. So these technologies that and probes, if you will, when properly calibrated and coupled with chemometrics, provide data, quality data for real-time release testing, either online or at line or inline. Much of this data that is collected by PAT sensors, however, do require a multivariate statistical analysis to relate the data that you have, to relate the numbers, to convert that into a practical understanding of the process. This sort of statistical analysis focuses on the relationships between more than one outcome variable. As major part of quality by design is design of experiments, which is really a systematic method that enables scientists and engineers to study the relationships between multiple input variables or factors and key output variables or responses. Basically, design of experiments is a structured approach for collecting data that attempts to maximize the amount of data you can obtain while minimizing the experimental work required. Obviously, design of experiment processes use some advanced multivariate statistical analyses to ascertain or understand which variables are the most critical and how these variables affect each other. In the next slide, I want to look at some of the common statistical methods that have been used during the implementation of process analytical technology. This slide shows the statistical methods and techniques that can be used in order to understand what your process analytical data is telling you. And I've listed a source here. There's an excellent article by Misra that goes into these methods in extreme detail, and getting too deep into these sort of spectral methods, pattern recognition is well beyond the scope of this talk, but I do want to focus on a couple of the most common, which are principal component analysis and partial least squares regression. These you keep seeing over and over and over again. They're constantly being used. They're very robust. And the problem that you run into is that data sets obtained from these spectroscopic tools are often very highly correlated or collinear with multiple other variables. So modeling a response with respect to a large number of, say, predictor variables can be difficult. And so data reduction and feature extractions methods become essential in order to shrink the data set down to something that's easier to understand. As an example, principal component analysis. A three-dimensional model of a shark allows the viewer to immediately identify it. But we can take a picture of a shark that's only two-dimensional, and we can still draw the same conclusion. In effect, it is still a shark. So in this example, we've been able to remove an entire dimension without affecting the conclusion. And this is really the power principal component analysis has. It works by focusing only on the most important aspects. What are the principal components? What are the primary drivers to allow you to discern a difference in the statistical analysis? And I do know that, yes, these are both two-dimensional pictures of sharks, but one kind of looks three-dimensional. But I think you can see the point. If you had a stuffed shark animal toy, perhaps, and you held it and you looked at it from all sorts of different angles, you're going to be able to tell that it's a shark. Now, if you just have a picture of a shark, it's still a shark. But of course, the angle of approach here is going to be different. You might not be able to tell in all situations, but principal component analysis does allow you to get rid of any redundancy, to reduce the data down to something that's much more manageable. And in a similar manner, partial least squares, which is kind of an extension of principal component analysis. It works in a very similar manner. Everybody uses ordinary least squares when you're doing something like linearity, or where you only have a couple of variables that aren't collinear. But as often is the case when these sort of advanced methods, collinearity is an issue that you need to overcome. In fact, it can be common. And these techniques of principal component analysis and partial least squares do provide for those issues to be overcome. And as I said on the previous slide, there are many others that have been used. And I think the Misra article is an excellent review of these techniques. And there are references that will be listed at the end of this presentation. So now that we understand patch sensors and we have an idea about the models that we're going to need to use with our patch sensors, these things need to be calibrated and validated against an external method. So you're going to need, when you're deciding on placement of process analytical probes, you need to make sure you've got some sort of a port nearby that you can actually remove a sample manually and confirm the result of the process analytical technology probe against, for instance, it might be an HPLC method or maybe a UV method. But the placement of those probes is a very important aspect that you really need to consider. Other considerations are probe type. Is it a retractable probe or not? Or is it just in situ continually? Placement, fouling of the probe, processing time. These are all big issues that you need to consider because the product flowing past the sensor window needs to be synchronized with the sampling frequency. If the wrong probe is selected, or if your placement is not optimized, retrofitting equipment and changing PAT probes can add exorbitant cost to the process. As an example, blend uniformity measurements. Usually you want to contain, or you want to measure those as close to the tablet press as possible, because you want to ensure that just prior to tableting, the blend was homogeneous. Several companies, and there are several process analytical probes out there that actually work in the feed frame of a tablet press. So you can get scans and be assured that the blend is uniform just prior to compression. You can also place some of these probes upstream to determine when mixing is complete. And a lot of times, if your control strategy requires data collection every 2 seconds, but the processing time takes 4 seconds, and that's not necessarily unusual, then you're going to need a different PAT tool or probe type. And often this can be discovered way too late, resulting in additional costs. And the bottom line here is that every process analytical technology tool is going to have risk, and the management of those risks should be part of your control strategy, and quality should be in on helping manage that sort of decision. Another issue that will crop up is the process analytical technology data management. These tools produce vast amounts of data, and all that data has to be captured, processed, interpreted, and stored, especially if you're going to use PAT data for release of product. You need to have that stored so that if regulatory agencies do an inspection, they can have access to that data to confirm that, yes, this product did meet specification. So there's a lot of technical skills that are needed here. Fortunately, computers have come a long way. We've got a lot more data storage capabilities. Some of the probes are usually under the control of a process analytical technology management software. And I've listed some examples here. I believe these are from GEA, the CintiQ and SciPat. And an important understanding, I think, here is that these process analytical technology tools convert spectral values into meaningful data that can be correlated with a critical quality attribute. The systems here that I'm discussing, they control data acquisition from the actual probes. The software also has to interface with multivariate statistical packages for model development, validation, etc. Um, a good thing about these systems is that they do allow for data mining, which can be very important in the validation and development of the statistical models that you will need in order to predict a critical quality attribute from the raw data. Process Analytical Technology Management System, the PAT management system, also has to interface with additional orchestration software to provide control over the entire system. And this is really mostly important from the standpoint of a continuous manufacturing environment, right? Integration into feed forward and feedback loops are necessary to ensure that any non-conforming material is segregated during the manufacturing process and doesn't ever make it out into the finished product. So the regulatory environment for process and analytical technology. Both the FDA and EMA, the European Medicine Agencies, have embraced quality by design and advanced manufacturing processes like continuous manufacturing. The FDA has actually created a group called the Emerging Technology Team, and their goal is to help industry with process analytical technology and quality by design in a continuous manufacturing process. Of course, this is highly beneficial for companies that wish to adopt these technologies because all the input from the emerging technology team is going to result in a faster review and speedier approval. And at the end of the day, the more quality you can show to regulatory authorities and the more control you can demonstrate over your process, the faster that is going to be approved. And then finally, a product's partial or total release is possible using process analytical technology data. Real-time release testing is not required when you use process analytical technology. So you can have process analytical technology, but not use that data to release. You can still take your samples and send them to the lab and go that way. However, it's unlikely that a real-time release testing could be implemented without process analytical technology. In fact, I don't see how you could if you have no data other than manual sampling. You're going to be hard pressed, which, as we've already discussed, the benefits here are faster time, speed to market, reduced analytical testing. Considerations regarding the cost of process analytical technology devices, equipment modification, process development, calibration model development, and validation of those models all need to be balanced against the savings that result from real-time release testing. And this will have to factor into your business system or business decision on whether to proceed or not. So, in summary, the implementation of real-time release testing requires business evaluation with a careful analysis of the cost, risk, and benefits. As part of the references, there is a wonderful paper by Markle that has an exhaustive review of the current status of real-time release testing in a pharmaceutical continuing manufacturing environment. And these are the references that I discussed. I have many more. Feel free to reach out to me via email, and I will be happy to share all the references I have with respect to this topic. I know we kind of covered a lot, but I really meant for this just to be kind of an introduction to the whole idea of process analytical technology, quality by design, and real-time release testing. So there's a lot more work that's out there that you can find. And as I said, feel free just to reach out to me if you would like additional references. Okay, I guess I can take some questions now. Is that right? Let's see. How CMA shall be determined and why? Is it mainly applicable for sterile materials? I'm not sure what CMA is referring to. I'm guessing maybe it's CQA. And that can be a complicated question. Mainly applicable to sterile materials. Yeah, that again, I'm not clear what that question is. What CMAs? I'm guessing maybe it's CQAs. Are these parameters applicable for only process validation or routine batch? Yes, they could be either or. As I specified, you could use process analytical technology not only for your process validation, but also for routine batch. And again, this kind of goes back to the idea of a quality by design. I mean, imagine a process where you can easily determine that every batch is blended to within a specific range. So you're essentially validating that every time. Is process analytical technology available for regulatory filings, and what is USP strategy on PAT tools, specifically on release of any general chapter? I will have to defer to the USP on what their strategy is on that, but process analytical technology is acceptable for regulatory filings. In fact, near IR spectroscopy, which is really, really a workhorse, actually there's its own guideline on submitting near IR data for process analytical technology. And it goes through exactly what you need to submit and how to submit it, what to do. It's a great guideline. Chemical small molecule. Yeah, you know what? I'm mostly talking about, in this case, small molecules, just because those are the easiest. But there are definitely a lot of research being done in both biologics and cell and gene therapy process analytical technologies. So I would expect you're going to see a lot more of those in the future. What is a maximum amplitude? You know, that's really difficult to answer. It really depends on the nature of the probe. As I mentioned before, some probes, such as the terahertz time domain spectroscopy, those have relatively long processing times. So that really, it's really impossible for me to answer that. Just the sampling frequency is going to depend on the exact type of probe you're using. And manufacturers of these process analytical probes are more than willing to help and work with you so that you choose the correct probe and you're placing it at the correct spot. Well, with respect to this, the next question, how could these techniques be applied to small scale, lab scale manufacturers in clinical trials? That is going to be difficult, right? I'm sure there are process analytical people that can design and help you implement a probe for any sort of scale, but for clinical trials, these are typically small batches, as you say, more lab scale. And my opinion is that in this case, you would probably just want to stick with your traditional form there. Although it's definitely worth reaching out to GEA and many other vendors uh, who may have alternative suggestions or ways to get around that. Okay, does PAT alleviate the need for a sampling fee? You still need this. Hopefully not. A sampling fee, of course, is just a way to take samples from the top, middle, and end of a blend, right? That's throughout some sort of cross-sectional area. And what you need to do when you're qualifying your process analytical technology is you need to have a sampling port where you can manually take a sample at the same time that the process analytical tool is monitoring to compare. Can critical material attributes be included into process validation batch as a parameter? One of the big things that has come out of the push for continuous manufacturing is that we're seeing a lot of companies that are actually developing databases of critical material attributes so that they can easily substitute one for another. Because as it turns out, the critical material attributes they can vary wildly, even maybe just from the same supplier, but from batch to batch. So it's important to include that and to think about that when it is appropriate. And is process analytical technology applicable for instrumental analysis? Process analytical technology needs to be validated against an instrumental method. But once you've done that and you've shown that I've got these two orthogonal methods that both give me the same number, then you can substitute your process analytical technology data for your real-time release data.